Hello everyone, myself Nainar Adhidya, and today I am going to talk or discuss about the intricate mechanisms behind the process of eukaryotic DNA replication. Let's begin. So, eukaryotic DNA replication is the process by which an eukaryotic cell duplicates its entire genome and passes it down to its daughter cell before the cell gets divided by cytokinesis. And we can divide this entire process into three phases: initiation, elongation, and termination. But before we dive deep into this process, I wanted to share a fun fact you can say. The overall error rate of this eukaryotic DNA replication process is roughly 10 to the power negative 8 to 10 to the power negative 10 mutations per base pair cell division. To put it into the perspective, we can say that roughly one wrong nucleotide is added for every 10 billion newly synthesized nucleotides. So we can say that this mechanism is so accurate that no human has ever created any kind of machine which can work at this accuracy or efficiency. And given below is an example of the librarian typo, which is classically used to explain the accuracy of the DNA replication process. So today I am going to give an overview about initiation, elongation, and termination phases of the eukaryotic DNA replication through this presentation. But the eukaryotic DNA replication process is much more than just these three phases. There is a process of epigenetic replication. If possible, then I'll also create a detailed video about it. So the entire initiation process or phase of the eukaryotic DNA replication process can be divided into these three major topics. First one is origin diversity, second one is ORC binding or origin recognition complex binding, and third one is pre-replicative complex assembly and firing. Before getting to know about origin diversity, we should have a basic understanding of the origin of replication or ORI. ORI is an essential DNA sequence where chromosomal DNA begins to unwind or melt and initiate the process of DNA replication during cell division. There are various complementary proteins are there in a cell which binds specifically to this ORI region in order to melt the DNA for initiating the process of DNA replication. In the case of prokaryotes, they possess a conserved protein known as DNA, whereas in eukaryotes, the most conserved protein is ORC, also known as origin recognition complex. Different organisms show variations in their origin of replication sequence in their genome, which arose from the evolutionary adaptations over generations of an organism. Thus, the type of origin which an organism possesses directly reflects the evolutionary lineage or adaptation of that particular organism. And if we compare, eukaryotes possess a significant variation within their ORIs when we compare it to prokaryotes. And this is majorly due to the complexity of eukaryotic chromosomes, which arose due to multiple factors like multiple linear DNA molecules, which are wrapped around the histone proteins, forming a complex structure called nucleosome, which are then further condensed in order to form a 3 nanometer chromatin fiber structure. And during the late prophase and or metaphase, the chromatin fiber condenses further even more in order to form an highly condensed structure of chromosomes or chromatids. This additional layer of organization necessitates more sophisticated mechanisms for various biological processes like the replication initiation, as well as coordination with various other processes such as transcriptional regulation and chromatin modeling, etc. Let's see what evolutionary flexibility does. The diversity in origin provides to an organism. The presence of diverse origins allows eukaryotic cells to respond flexibly to environmental stresses or mutations that may affect replication machinery components. And the evolutionary flexibility can be explained in two with respect to two factors. First one is stress response, and second one is mutation tolerance. So, in stress response, a organism with a variety of active replication origin can better tolerate DNA damage by activating alternative pathways for genome duplication, which helps maintain genomic integrity and cell viability under adverse conditions. We can understand this point with an example of, suppose there is an specific type of yeast. If we put that yeast in an harsh condition, what it will do that it will try to suppress its ORI sequences and the ORC complexes, or the gene which codes for the ORC complexes, thus stopping the process of DNA replication, and it will not divide until an favorable condition occurs. So, if an favorable condition comes, the yeast will upregulate the genes which code for ORCs and removes any suppressive factors which act on the ORI sequences, thus increasing its growth rate and dividing rapidly. This mechanism allows it to survive better in various or changing environments. On to the second point, which is mutation tolerance. The redundancy provided by multiple origin of replication can also buffer the effect of deleterious mutations affecting despite genetic alterations that might otherwise compromise genomic stability or organismal fitness. For an example, there are five origin of replication in this given image. If any kind of deleterious mutation happened in three of these regions, then other two can act as the origin of replication. Thus, there will be much less. Thus, you can say even if you have multiple deleterious mutations in multiple origins, even if there is one or two uh, functional origin of replication, the DNA replication process will proceed without any interruption. Here, the term redundancy means there are more in number than we need. That is, in context of origin, we only need one or two, or you can say according to the length, we will need approximately, suppose there is 100 nucleotide long DNA strand, then we will need approximately one or two origin will be more than enough. But a cell will contain 5 to 10 origin of replication for that. This is an example of redundancy. Here are some key points that highlight the diversity in eukaryotic origin of replication. The first point is the number, arrangement, and sequence specificity. Typically, eukaryotes have multiple ORIs scattered throughout their genome to accommodate large genomes with extensive non-coding regions. And they also serve a different purpose or many purposes. One among them is the redundancy factor, which we discussed in our previous slide, that if an ORI contains or gets an deleterious mutation through which it won't doesn't able to initiate the DNA replication process, and another ORI sequence takes its place and initiates the DNA replication process. Here's an example that humans possess tens of thousands of potential origins that are regulated and utilized at different times during developmental stages and cell types life cycles. We will discuss this example in more detail in the fourth point, that is cell type specific origins, differential expression, and regulation. For now, let's get into the second point, which is the nature of ORI sequences. In prokaryotes, when we compare prokaryotic ORI and eukaryotic ORI is far more complex than prokaryotic ORI, where prokaryotic ORI contains two separate sequences, 13-mer and 9-mer, which one contains 88 sequence and other contains the GC sequence, where the DNA protein binds and starts to melt the DNA. Whereas in eukaryotic ORI sequence, we can classify into the ORI sequence in two groups: metazoic ORI of metazoans and ORI of non-metazoans. Where metazoans are those organisms which are complex multicellular organisms like humans, where non-metazoans are yeast-like organisms which are far simpler than the uh, metazoans or multicellular organisms. So in non-metazoans like yeast, they have a highly conserved ORI sequence which is known as ARS, which stands for autonomously replicative sequences. But in non-metazoan or sorry, in metazoans like humans, we have a very complex ORI sequence, and we don't have any conserved specific sequence. The only thing we know about the ORI sequence of metazoans is that every ORI sequence of metazoan share a commonality. That is, they contain 80 regions, CPG islands, G quadruplexes, or specific motifs like TSS. They also contain nucleosome-free regions, etc. But there's a catch that uh, it's not mandatory that every ORI should contain all of this. The meaning is that every ORI of eukaryotes contain any of these factors. Like some ORI contain only ATG rich regions. Some ORI may contain CPG islands and nucleosome free region, etc. These variations reflect differences in how different organisms have evolved mechanisms for initiating DNA synthesis from these origins, while avoiding unwanted activation at non-replicative sites, which can lead to genomic instability. The third point is organismal complexity. The scale at which eukaryotic cells operate implies a higher demand for accurate DNA duplication to maintain multicellular organization. That is, uh, a multicellular organism contains different types of cell or differentiated cell which contains same genome but has different structure and performs different functions. Thus, the ORI machinery needs refined precision in targeting origins within chromatin context, that is the epigenetic regulation, uh, which includes methylation, acetylation, histone modification, or modified histone complexes like H31, etc. A level of control and regulation not typically observed in prokaryotes. Now, the fourth point is cell type specific origins, differential expression, and regulation. Different cell types express distinct set of origins which contribute to their unique patterns of gene expression during development and differentiation process. When we combine these example and this example, we can get a clear picture. So, as we know that these stem cells are a specialized cell which can replicate or perform cell division uh, at an extremely high rate, even though stem cells and normal eukaryotic cells contain the same copy of genome. But the difference comes that the stem cells contain a large number of active replication origins, that is, which initiates the DNA replication process, whereas normal eukaryotic cells contain a comparatively small number of active replication origins. This is a regulatory mechanism which helps the normal cell to not become any cancerous cell. Thus, only stem cell contains a very large number of active replication origins. But when in another case, that is the case of terminally differentiated non-dividing cells, for an example is the developed neurons, which don't divide, uh, have practically useless ORI, or their URI doesn't serve any purpose because they don't divide. And from this slide onwards, we are going to understand about the actual machinery or mechanisms behind the eukaryotic DNA replication process. But before understanding the ORC binding or origin recognition complex binding, we should have a basic understanding about the structure and function of the ORC complexes in eukaryotic cells. As I mentioned previously, that the ORCs are a complex of six different proteins which are named as ORC1, 2, 3, 4, 5, and 6. Thus, they make an heterohexameric complex where which comes together and forms an ring-like structure which binds specifically to the ORI sequence in the genome. And the process of binding of ORC complexes to the ORI sequences occurs only in G1, late G1 phase in the cell cycle of eukaryotic cells. As we know that eukaryotic cells life is can be explained by a life cycle which contains primarily four phases. First one is G1, S, G2, and M phase, and in some cases G0 phase. So this process, that is ORC binding with ORI sequence, occurs only in G1 phase, that is before cell division begins. And if possible, we will discuss about the entire regulation of the DNA replication process in eukaryotes in context of cell cycle in a separate video. But for now, just remember that the eukaryotic DNA replication initiation step, the ORC binding with ORI sequence only occurs in the late G1 phase. As I mentioned that these proteins, this is an hexameric ring protein which contains six different subunits, namely ORC1 to ORC6, which recognize the DNA sequence at ORI sequence. But the interesting part is these proteins, through their conserved domains such as ATPase and helicase and CDC6P binding sites within ORC3 and ORC4 respectively. That is, as I mentioned, that this is a complex protein which contains six different proteins in them. And among them is ORC3 and ORC4, which contains ATPase and helicase domain and CDC6P binding site, where CDC6 stands for cell division cyclin 6P. It is an chemical messenger, you can say, which is also used in the regulation of the entire DNA replication process, and it also plays a crucial role in the actual binding and stabilization of the ORC protein with the ORI sequence. So ORC serves two important functions. First one is they mark the origin regions for subsequent assembly of pre-replicative complexes. That is, there are many proteins which comes together in order to form a pre-replicative complex, which then initiates the DNA replication process by recruiting other replication factors such as this CDC6 and the CDT1. Additionally, they actively participate in unwinding DNA to facilitate accessibility. That is, they also participate in the process of DNA melting in the initiation step. That is, in initial phase of the initiation process, they also help to melt the DNA so that the DNA can, or I can say that we can say that the helicase protein and other proteins such as polymerases and single-stranded binding proteins can come together and start proceeding further. And now uh, let's talk about the variations across eukaryotes uh, in context of ORCs. While the core components of ORC are conserved across eukaryotic species, 1 to 4, variation exists that reflect adaptation specific to different organisms or cell types, such as differential expression levels and post-translational modifications. These differences contribute to a diverse range of replication initiation mechanisms in various lineages beyond East. So, for fun fact that the origin recognition complexes were first discovered in the yeast cells, and also that there is an difference between the structure or protein structure of metazoan ORCs and non-metazoan ORCs. So now let's discuss the similarities and differences between metazoans and non-metazoan origin recognition complex binding diversity in eukaryotic origins of replication. So this paragraph is just an revision of what I mentioned earlier. So firstly, the similarities between metazoan and non-metazoan origin binding are that they have or they share a common core set of proteins, replication proteins like ORC, CDC6P, CDT1P, and MCM227 complex. These conserved components are essential for the assembly of pre-replicative complexes at replication origins across all eukaryotes studied till date. Yeah, till date. The first point is conservation across domains. So the presence of the conserved domain like the ORC protein core suggests that there is a shared ancestral origin highlighting their fundamental role in DNA replication initiation. This commonality underscores the importance of maintaining genome integrity through precise regulation across diverse eukaryotic lineages. That is, these conserved domains signify that the metazoans and non-metazoans share a common ancestor which possessed these core protein domains for commonality. Thus, there is thus these commonalities are conserved in both metazoans and non-metazone creatures. Now, second point is functional similarity. Despite evolutionary divergence, both metazoans and non-metazones utilize ORC proteins to recognize specific DNA sequences, that is ORI, at replication origins, facilitating the recruitment of additional factors required for pre-assembly, pre-RC assembly, and activation. This functional similarity emphasizes that core mechanisms governing genome duplication are preserved across eukaryotic domain, even in organisms with vastly different histories and cellular architecture. So both of these topics or points have been discussed in detail in our previous slides. So let's jump to the differences between the metazone and non-metazone origin binding. So the first difference is sequence specificity. As I mentioned earlier, that the metazone features have a conserved ORI sequence, which is known as ARS, which stands for autonomously replicative sequences, whereas non-metazoans have uh, comparatively complex origins which have no conserved sequences. This variation suggests that evolution has fine-tuned the recognition compatibilities of ORC proteins in different eukaryotic lineages, potentially influencing replication, timing, frequency, or regulation within specific genomic regions. So, the regulatory mechanism, or the second difference is regulatory mechanisms. So, non-metazoan organisms such as yeast often display simpler regulatory networks governing DNA replication initiation compared to metazoans with their more complex cellular differentiation process and tissue organization requirement. As we know that the metasome features like humans have differentiated cells, even though each cell contains the same genome or same copy of genome. Thus, we need a far more complex regulatory mechanism in order to control the protein expression level and gene expression level, etc. For an example, there is an ubiquitination process happens in the termination process, which we'll discuss in detail uh, in the MCM27 complex. Now, the third point is evolutionary pressure. The divergence observed between metazoan and non-metazoan ORC binding likely reflects the distinct evolutionary pressure faced by each organism, such as adaptation to different environmental niches or life cycle strategies, unicellular versus multicellular. These adaptations may have driven changes in replication origin, organization, and regulation that confer advantages for survival and reproduction within specific ecological contexts. This is an illustration of how an origin recognition complex, that is 6-hexameric ring protein, binds to the ORI sequence, and here is CDC6 protein, CDT1, and this is the hexameric helicase, that is MCM27 complex. So now let's try to understand about the pre-replicative complex assembly. When the ORC binds to the ORI sequence, as shown in the figure or illustration in the previous slide, it recruits several other proteins and complexes like CDC6, CDT1, and MCM27 helicase, where MCM stands for mini chromosome maintenance protein, and 227 is the name of different proteins which comes together in order to form this helicase complex. Just like in the case of ORC, there are ORC 1, 2, 3, 4, 5, and 6 proteins. In the case of MCM27 helicase complex, there is MCM2, 3, 4, 5, 6, and 7. This is also an hexameric complex during G1 phase. So during G1 phase, the pre-replicative complex formation takes place, and only in G1 phase the pre-replicative complex form assembly takes place in order to prevent multiple duplications of the DNA. And the process of pre-replicative complex assembly is known as licensing event or DNA replication initiation licensing event. And the complete replicative complex will have MCM27 double hexamer. That is, there will be two helicase MCM27 helicase complex encircling the DNA. So this is a glimpse of how the DNA replication process is regulated. That is, the cell cycle checkpoints ensure that all necessary conditions are met before proceeding with each phase. As we know that in cell cycle, there are three checkpoints where The cell ensures that there is no anomalies in its uh, processes and its composition. That is G1 S phase S checkpoint checks whether every protein and nucleotides required for the DNA replication process is there or not. And in G2M checkpoint, the cell ensures that the DNA has no or very minimal DNA mutations after the DNA replication process. And in mitosis spindle fiber checkpoint, the cell ensures that the splitting of the chromatid sister chromatids has taken place properly. So let's understand the different aspects of the pre-replicative complex in detail. So the first point is origin recognition complex formation or ORC formation. I have explained these 3 points in detail in our previous slides. So, but you will see a new term which is origin fires or origin firing. We will discuss about this term in detail in our upcoming slides. Origin firing. So just remember this for now. And as I mentioned earlier, that ORC serves as an scaffold recruiting other factors necessary to form the complete pre-RC structure and initiate DNA replication. For example, MCM27 helicase complex, CDC6, CDT1, etc. So now let's jump into second point, which is CDK activity regulation, where CDK stands for cyclin dependent kinase. These are a class of kinase enzymes. Kinase enzymes are those enzymes which can phosphorylate a given substrate. So, cyclin-dependent kinase refers to a class of enzymes which can only perform its kinase activity when bound to uh, a molecule or a cofactor known as cyclin. In cell cycle, there are many CDK and cyclin pair play a role in many different uh, processes, and one among them is CDK2 and cyclin E during late G1 phase. So what they do is they phosphorylate specific units of ORC so that they once an ORC and the origin recognition once the ORC and origin binds together, so the CDK2 and cyclin E comes and phosphorylates the ORC complex in specific subunits. This prevents the reassembly of any another ORC uh, in any other origin, so that the replication initiation occurs only once per cell cycle and is tightly controlled in time with other mitotic events. But the CDK activity must be down-regulated to allow the reassembly of origin recognition complex at origin for a second round of replication initiation during late NG1 transition. This is achieved through phosphorylation of other kinases such as DDK kinase and CDC6, MCM27 complexes, CDT1, etc. We'll discuss about this in much more detailed manner in another video, which focuses only on the regulation of DNA replication. So the assembly of pre-RC or pre-recognition complex is mediated by cyclin-dependent kinase, and there are also several other proteins which work in concert with ORC in order to form this pre-RC complex. This given infographic is all you need to know about for now about the pre-recognition complex. The pre-recognition complex is mainly formed by ORC, CDC6, CDT1, and MCM27 complex. These four protein and complex interact with each other in order to form this pre-IC or pre-initiation complex, which leads to the formation of open seal conformation of MCM27 helicase, which only binds to a double-stranded DNA. We'll talk or I'll explain about this open seal conformation later in this video. So the primary work of these two proteins, which is CDC6 and DPB19 or also known as CDT1, is to help load the MCM helicase complex onto the ORC complex and stabilize them through ATP hydrolysis and phosphorylation activity. The CDK dependent or cell cycling dependent kinase is also uh, primarily needed to load and activate the MCM helicase onto the ORC complex. Let's talk about origin activation or firing step. This is the phase of DNA replication initiation process where the origins finally get activated or where the DNA replication of eukaryotes actually begin. Here, the primary motto of this process is the formation of CMG helicase, which stands for CDC45, MCM27 complex, and genes. These three proteins and protein complexes come together to form this CMG helicase, which is an functional helicase, which is what actually denatures or melts or unwinds the double-stranded DNA for the process of DNA replication. So, so let's jump into the detail. The definition is origin firing is the process by which a pre-RC transition from an assembly state to active participation in DNA synthesis, marking the beginning of chromosomal replication at specific sites called origin. This event involves several steps in molecular prayer. So, in this step, the replication fork establishment takes place, or you can say replicosome or replication bubble formation takes place in this step, that is origin firing step, where the CMG helicase melts or unwinds the DNA for the elongation step, thus forming the replication fork or replication bubble. In this step, DNA polymers also get recruited by CNG helicases and PCN and other factors, which we'll discuss uh, later in this video. As I said, given in this infographic, we can say that CDC45 genes and MCM27 comes together and makes up the CNG helicase, which has a closed seal conformation, where MCM only binds to single-stranded DNA, which we'll discuss in more detail. So these are some auxiliary mechanisms or processes which help the formation of CMG helicase. First one is CDC6 activation. The activation of CDC6 by phosphorylation marks a critical step towards origin firing, as it allows MCM helicase loading onto the DNA, which is caused by the ECDK2 cyclin ECDK2 pair. The next one is MCM activation, which is also mediated by the CDC6 phosphorylation event, which triggers many cascade of reactions like uh, DBF4 dependent kinase, DDK kinase, which is a type of kinase which phosphorylates some specific subunits of MCM complex, in turn activating the MCM complex. Then comes the origin recognition complex phosphorylation, which is also phosphorylated by some CDK kinases, is an another crucial step that promotes the transition from pre-arch assembly to origin firing, as it prevents assembly of MCM complexes at origins and allows for replication initiation, which we have discussed in our previous slide. Then comes genes assembly. Genes are a group of four different proteins, which are namely SLD5, PSF1, PSF2, and PSF3. Well, the gene's name was given by a Japanese scientist who discovered these four proteins, where genes um, stands for Go Ichi Ni San, which is an Japanese term for Go stands for 5, Ichi for 1, uh, Ni for 2, and San for 3. It symbolizes the suffixes of these protein 5, 1, 2, and 3. This assembly is primarily responsible for recruiting proteins like RFC, PCNA, and also uh, in the process of changing the MCM complexes open or open seal to closed seal conformation. CDC45 complex is a heterodimer that binds to single strand of separated parental DNA. It also helps in loading of MCM 2 to 7 complex. Also, this also plays a crucial role in the conformational change of MCM complex from open to closed seal conformation with genes assembly. And some other proteins which gets recruited are PCNA, RFC, RPA, DNA pools, where PCNA stands for proliferating cell nuclear antigen, RFC stands for replicative factor C, RPA stands for replicative factor A, and DNA pools are polymerase. Now let's try to visualize the DNA replication initiation phase, which we have learned so far. This is this is the ORI sequence where an ORC or origin recognition complex comes in, recognizes it, and binds with it. Then it recruits CDC6 cell division cyclin 6 and CDT1 uh, one protein which uh, binds with the ORC or origin recognition complex and stabilizes it and stabilizes the overall complex, which then in turn recruits MCM27 helicase, which is then binds either side of this complex, thus forming what is known as the uh, helicase double hexamer, where we have two helicase hexamers. Thus, we refer to it as helicase double hexamer. And as we have learned that here MCM27 helicase is an hexamer which contains six different subunits MCM2 to 7. Here is what we mean by that. Now let's talk about open seal conformation. When we use the term open seal conformation, we refer to this. This is what we refer to as open seal conformation of MCM27 helicase, where this is the MCM2 subunit and this is the MCM5 subunit. And the double stranded DNA is enclosed like this is how the open cell conformation MCM helicase encloses the double stranded DNA. But this conformation is unstable, thus it cannot unwind the double stranded DNA. Thus, the pre-initiation complex step comes in, where the double hexamer open cell conformation DNA is open cell conformation complex is here. Then two kinases are recruited. First one is DDK, but before that, this is what we refer to as the pre-initiation complex, and this is formed during the late G1 phase. And in licensing event, this is what we meant by licensing event, where the pre-initiation complex is bound with the ORI region. This is what we call origin licensing, where the pre-initiation complex is bound with the origin of replication. Now, let's continue. When the DNA licensing has happened and the synthesis phase or S phase of the cell cycle starts, some kinases are recruited. Among them are DDK and CDK, where DDK stands for DBF4 dependent kinase and CDK stands for cyclin dependent kinase. They phosphorylate CDC6 and CDT1 and also phosphorylate the MCM helicase, which changes its conformation to the closed seal conformation, which looks like the closed seal conformation looks something like this, which is only bound to one single strand of DNA, which is like this. And in this form, the helicase is active and it can easily unwind the DNA. And after this, but in order to uh, create this stable closed seal conformation, some protein factors are needed, which are genes and CDC45. And let me show you how it will look like when it binds to the MCM27 helicase. The complete structure of closed seal conformation MCM or MCM complex will look something like this, where this is the MCM 27 complex. This is the second subunit and this is the fifth subunit. This one is the genes and this one is CDC45. This and this has now a new name, which is called CMG helicase, which stands for CDC45, MCM27 and genes. This is what we refer to as CMG helicase. This is the structure, how it will looks like. The CDC45 and genes uh, fill the gap between the 2,5 subunits of the MCM27 helicase, thus forming the closed seal conformation like this. After this, the phosphorylation event takes place, which activates the helicase. After the activation of helicase, auxiliary factors like polymerases and tCNA, uh, that is beta clamp loader and beta clamp single stranded binding proteins, also get recruited at the complex at the repliosome, and this is what we refer to as repliosome or replication bubble. These auxiliary factors gets recruited, then uh, the process of uh, DNA replication, eukaryotic DNA replication, or to be precise, elongation step proceeds like this. And this step where the closed seal conformation has been obtained or CMG helicase has been obtained is what we refer to as the origin firing. In origin firing, we will have the closed seal conformation or CMG helicase, which is now active and can unwind or melt the DNA. Now let's move forward to elongation step. Now the next phase in eukaryotic DNA replication is elongation, and the elongation can be divided into three portions: bidirectional force, primary DNA polymerase, and processivity factor. In bidirectional force, we will understand the dynamics of the uh, force, replication force in the replication. In primary DNA polymerases, we will understand three major polymerases: DNA polymerase alpha, delta, and epsilon. And in processivity factor, we will discuss about the protein factors which ensure that the DNA replication process or elongation step has maximum efficiency and accuracy. During the elongation phase of the DNA replication process, a crucial aspect is that the synthesis proceeds from two opposite directions on each newly forming daughter strand. This process creates what are known as bidirectional forks because they generate two leading and two lagging strands at both ends, that is forward and reverse, simultaneously within the same origin region. Uh, as you can see in this figure, this is what we call as a replicosome or replication bubble. And if we divide it in half, then we will create two replications. One, first one is this, and the second one is this. And as you can see that in both the replication forks, the elongation process is occurring simultaneously on both the ends, that is on the upper strand and on the lower strand. And this generates two leading strands and two lagging strands. If we consider that this is the 5 prime end and this is the 3 prime end, then the DNA pole will synthesize 5 prime to 3 prime in this direction and creates an leading strand. And from this direction, that is uh, 5 prime to 3 prime direction of the parent strand, the DNA synthesis will occur in fragments of which we will refer to as lagging strand, and thus in a single parent strand we can see both leading and lagging strand. If this is 3 prime and this is 5 prime end of the parent strand, then the synthesis from 3 prime end to 5 prime end of the parent strand will be leading strand, which means the DNA, the newly synthesized DNA will be from 5 prime to 3 prime in this direction. And when we consider the 5 prime to 3 prime direction, the newly synthesized strand will be discontinuous and thus known as lagging strand. And the same case is applicable to the bottom strand, which is where the directions will be opposite but the concept will be same. In bottom strand, this will be the leading strand synthesis direction, and this will be the lagging strand synthesis direction. And thus, we refer to this model as bidirectional forks, where we have um, where a single fork will have two directions of synthesis. And bidirectional fork progression is influenced by several other factors, including nucleotide availability, replication stress responses like SOS response due to DNA damage, and cell cycle checkpoints that modulate the assembly of pre-replication complexes at origins during interphase or inactive them under adverse conditions such as starvation for essential nutrients like nucleotide precursors, etc. So, the regulation happens like if some mutation occurs, then the entire replication bubble or replicosome will be halted for certain movement so that the DNA repair mechanism can come in and fix the incorrect nucleotide. Let's move on to the next question, which is DNA polymerization. Before moving on to the DNA polymerase topic, let's discuss about some key features involved in the bidirectional force dynamics include. Uh, the first 2 points are pretty basic. So let's go through quickly. First one is the leading cell synthesis, which is continuously towards the replication growth because DNA polymerase can only add nucleotide on an existing 3' end. Therefore, they utilize a template that is oriented towards the direction of 5' to 3'. This process ensures high efficiency and speed in duplicating genetic information. Where let if I give you a biochemical perspective of this particular process, so what happens is the 5' phosphate end of the three nucleotide performs an nucleophilic attack on the 3' hydroxyl group of the existing primer nucleotide, which creates the phosphodiester. Then comes the ligand strand, which are synthesized discontinuously away from the replication source due to the anti-parallel structure of DNA. Short fragments known as Okazaki fragments form, each requiring priming by RNA primer, binding followed by elongation and eventually ligase joining them into a continuous chain. The third point we will discuss in more detail in the topic of lagging and leading strand coordination. But if I give a brief that the uh, Okazaki fragments are removed or RNA primer are removed by these three, primarily by these three enzymes. When one nuclease, RNase, H and DNA fold, and the gaps are filled by DNA polymerases such as DNA polymerase delta and the uh, single-stranded breaks or nicks which are created after the activity of DNA polymerases are sealed by DNA ligase which maintain the genomic integrity. This synchronization prevents conflicts at stalled forks where one strand might be synthesized but not properly repaired or completed on the other due to fork progression issues such as tension caused by unwinding of superpulse ahead of Newton flow, which can halt polymerases and cause breakages. Well, this particular problem, which is unwinding of coils ahead, creates a tension which can halt DNA polymerases and causes breaks. Can be is or should I say that is solved by the topoisomerase enzymes, which generates negative supercoils in order to uh, lower the or release the tension. It does this by creating single-stranded or double-stranded breaks on the DNA ahead of the ahead of the supercoil. Then comes the bidirectional nature or this point which I have discussed in our previous slide of DNA initiation step. Now let's understand about the primary DNA polymerases which are utilized in the process of eukaryotic DNA replication. Well, DNA polymerases are highly diverse enzymes that evolved from different origins, like from viruses, we have viral dual transcriptases. The human version is the telomerase, which also possesses reverse transcriptase activity. They perform various functions and uh, some of them are repair mechanisms and the actual DNA replication process, etc. Their activities are modulated by a variety of factors such as post-translational modification by phosphorylation, protein interaction with other replosome components involved in DNA synthesis, initiation elongation mechanisms like PCNA and RPC. Their structure reveal different conformations during various stages of replication processes that reflect the dynamic nature of these enzymes and how they interact with other proteins to achieve precise coordination within eukaryotic cells machinery for maintaining genomic integrity. So before understanding each type of primary DNA polymerases, this is an let's understand about or let's try to solve this question, which states that why does only RNA polymerase are able to synthesize polynucleotide chain from scratch without needing uh, or without the need of free 3' OH end, whereas DNA polymerase needs a 3' hydroxyl group in order to synthesize its DNA. As I have mentioned that the 5' group of the new nucleotide uh, performs a nucleophilic attack on the 3' hydroxyl group of the existing DNA primer, and this exact nucleophilic attack is catalyzed by the DNA polymerase enzyme. So, I would encourage you to pause this video in here and think about this question, that why does only RNA poles are able to synthesize polynucleotide from scratch without the need of 3' hydroxyl end. So now let's see the answer, which is according to the RNA world hypothesis, due to early arrival of RNA polymerases, they are comparatively more advanced than any other DNA polymerase, which means that even before DNA existed, RNA was used as the genetic material by many different organisms. Thus, the uh, RNA polymerase enzyme were existed way before the existence of both DNA and DNA polymerase enzyme. Thus, the RNA polymerase had time and no competition, had enough time to evolve some sophisticated mechanism or specific mechanisms to synthesize RNA or polynucleotide chain from scratch, and it doesn't have any uh, auxiliary mechanisms or helping mechanisms which help it in the process of the synthesis or replication. But on the other hand, when the uh, DNA became the genetic material of many organisms and DNA polymerases evolved, they had a substitute, which is RNA polymerase, which can synthesize RNA from scratch. And there are some RNA polymerases like primases and pol alpha, which uses DNA as template, reads DNA, but synthesizes our complementary RNA. And DNA pole can utilize the free 3' OH of the primer to extend the or synthesize the DNA. Thus, only RNA poles have the ability to synthesize the polynucleotide chain from scratch. Now let's move on to every type of DNA polymerase which are involved in the eukaryotic replication process. Now let's discuss about every type of DNA polymerases, major DNA polymerases which are utilized in the DNA replication process in eukaryotes. First one is DNA polymerase alpha. The role of DNA polymerase alpha is the initiation of DNA synthesis by creating an RNA-DNA hybrid, that is, an RNA primer which binds to DNA that serves as a starting point for replication at origin of replication sites within chromosomes. That is, the RNA primer provides the free 3' hydroxyl group to the DNA pole delta and epsilon. It possesses both priming and elongation capabilities, which are essential during early stages of replication when Okazaki fragments on the ligand strand need to be synthesized by other polymerases later in test phase. The mechanism behind is pretty interesting if we see. The whole alpha has four subunits: P110, P60, sorry, P180, P68, P58, and P48. Amongst them, P118, P68 specializes in DNA synthesis and RNA synthesis respectively. That is, the P180 synthesizes DNA and P68 synthesizes RNA, and others play role in interaction and regulation, which means the P58 and P48 subunits interact with uh, com- other reprosome compounds like PCNA and RPCs in order to increase their efficiency or processability. It is a part of a larger complex that includes PCNE, that is proliferating cell nuclear antigen, as its sliding plan, which increases the processivity during DNA synthesis, as I said before. The distinctive feature which separates it from other DNA poles is, unlike other polymerases involved in replication, pole alpha has a unique ability to initiate DNA synthesis without pre-existing primer. This is because it has higher affinity towards RNA-DNA hybrid than double-stranded DNA, which means the uh, P180 subunit and P60 subunit has higher binding affinity towards the RNA-DNA hybrid than the double-stranded DNA itself, due to its intrinsic preference of DNA type A. As we have an idea about the different types of DNA, that there are majorly 3 to 4 types of DNA: DNA A, B, uh, C, D, and Z, where DNA B is the most prominent one and is mostly found in all organisms, uh, which we mostly study about. But the DNA type A has an interesting, what we can say, structure due to hydrogen bonds, which is similar to the RNA DNA hybrid. Thus, the DNA polymerase alpha tends to bind RNA DNA hybrid rather than DNA DNA hybrid. Thus, what happens is, after synthesizing uh, 10 to 15 nucleotide of RNA uh, primer in the template strand by using its P118 P60 subunits, what happens is, as it slides through the uh, double-stranded DNA and the processivity of the uh, RNA polymerizing subunit, which is P68, is comparatively very low because it only creates 10 to 15 nucleotide of RNA prime. So what happens is, suppose this is the 10 to 15 nucleotide RNA primer, and the polymerase moves in this direction. Suppose. So what happens is, if the RNA polymerase moves in this direction, the P180 subunits gets activated after some time, as the P68 has a very low processivity. So the P180 subunit takes over and creates double-stranded DNA. And what happens is, as it has very low affinity towards the double-stranded DNA, it just falls off the double-stranded DNA after some time. This is due to it also lacks very crucial mechanisms, which is proofreading mechanism. Unlike unlike other DNA polymerases, it doesn't possess proofreading ability, which means if it mistakenly puts any wrong nucleotide, it can't proofread and remove it and fix the problem. It just goes on and synthesizes even if the DNA contains much more error, or we can say mistakenly put wrong nucleotide. Thus, it falls off after some time. It after approximately, you can say, 10 to 20 or 20 to 50 nucleotides of double-stranded DNA synthesis, which means after creating 10 to 12, 10 to 15 RNA primer nucleotides, it creates DNA, double-stranded DNA with its P180 subunit. Then after creating some uh, approximately 50 to 100 nucleotide length short DNA fragment, it just falls off. Its regulation is done by, along with other replication factors such as CDK, cyclins, and ORC proteins, is tightly regulated to ensure that DNA synthesis occurs only at appropriate stage, and the coordination ensures genomic integrity by preventing re-replication of unscheduled replication events. But the primary reason for this specific polymerase regulation is to not create a DNA which has multiple errors in it. The primary interaction of alpha polymerase alpha is with CMG complex, TCNA, and some ORC complex, which it gets loaded in the beginning of the initiation process or at the end of the initiation process. Then, what are its implications? Mutations in polymerase alpha can lead to genomic instability, contributing to diseases such as cancer. Its role is also critical for maintaining telomere length, although through the synthesis of short RNA primer at each and during replication initiation by polymerase alpha, which we will discuss later in this one. Now, the next DNA polymerase which we are going to talk about is DNA polymerase delta. The primary responsibility for this polymerase is DA is lagging strand synthesis and also involved in Okazaki fragment process. DNA polymerase alpha is an high fidelity enzyme, which means it has an high processivity. It can synthesize long strand of DNA without falling off. That ensures accurate replication of the bulk of eukaryotic genomes. It has proofreading ability to correct errors during elongation. But we cannot say that only DNA fold delta is able to synthesize the lagging strand in DNA replication, as we have evidences that um, or which shows that sometimes DNA epsilon may synthesize the lagging strand and sometimes DNA fold delta may synthesize leading strand. That's why here I have wrote that its primary responsibility is to synthesize lagging strand. Now let's get into the mechanism. Uh, DNA fold delta possesses both exonuclease and polymerase activities, allowing it not only to synthesize DNA but also to remove incorrectly incorporated nucleotides, which is proofreading ability, which DNA fold alpha lacks. That's why this enzyme can uh, synthesis long stretches of DNA with very minimal error. Its processivity is further enhanced by the TCNA sliding clamp that encircles the DNA strand, which I will show later in this video. And then comes the distinctive feature, which separates these enzymes from the other two DNA polymerases. Pol delta has high affinity for Okazaki fragments and plays an essential role in their processing, including the removal of RNA primers by its intrinsic exonuclease activity. That is, when the DNA pol delta synthesizes the lagging strand, it also has the ability to uh, remove the RNA primers using its exonuclease activity and synthesize the complementary DNA, thus filling the deoxyribonucleotide gaps caused by the primary. This also involved in the DNA repair mechanisms such as base excision repair due to its ability to remove damaged bases from the template strand during replication using its exonuclease activity. Now let's talk about the regulation of DNA fold delta. The expression of DNA fold delta, like that of other formulations, is regulated by the CDKs, cell cycle dependent factors including CDKs and cyclins, which ensure proper timing, at, which we have discussed in our previous slides. Its activity can be modulated through post-translational modifications such as phosphorylation. When so sometimes what happens is when we some enzymes phosphorylate these sub phosphorylate some subunits of the whole delta, which can upregulate or downregulate the DNA replication process or even halt the DNA replication process. Now comes the interaction. DNA pole interacts primarily with pCNA in order to increase its processivity. Then RFC complex that loads the sliding clamp onto the DNA during initiation and elongation uh, step. Then comes FEN1, that is flap endonuclease 1, which interacts with polymerase delta in order to remove any RNA primers or RNA primer flaps generated during the replication process, which we will discuss a bit later in this video. And other factors involved in coordinating lagging strand synthesis such as CFT, CTF, CSTF. Now the implications are defects or mutations affecting DNA pool delta can lead to genomic instability due to incomplete replication of the genome, contributing to various diseases, including cancer. It is also implicated in aging and cellular senescence as a result of slowly maintaining telomere length through Okazaki fragment processing at chromosome ends during DNA synthesis initiation by pole alpha, which we'll discuss in the topic of end replication problem and telomeres. Now let's move to the final DNA polymerase, which is DNA pole epsilon.
Now let's talk about DNA pool epsilon, whose primary role is to synthesize leading strands. But just as the case of DNA pool delta, we also have evidences which suggest that DNA pool epsilon sometimes synthesizes lagging strand also. Thus, when we talk about the role of DNA pool epsilon, we often refer to it as the primarily synthesis of leading strand, not only the synthesis of leading strand, and the association with the PCNA complex forming a polymerase complex that ensures efficient and accurate DNA synthesis on this template strand. Now, when we talk about mechanism, the pole epsilon has the highest processivity among the three primary DNA poles, that is DNA pole delta, alpha, and epsilon, meaning that it can add thousands of nucleotides without dissociating from the DNA template. This is facilitated by its interaction with pCNA through a clamp promoter complex that opens and inserts pCNA onto the DNA strand before recruiting pole epsilon to initiate replication. That is, DNA pole epsilon already has an high processivity, but with the interaction or association with pCNA complex increases its processivity several folds drastically. And also, DNA pole epsilon possesses proofreading activity. That is, it has an intrinsic 3' to 3' to 5' exonuclease activity, which allows it to remove incorrectly incorporated nucleotides. That is, it removes incorrectly incorporated nucleotides immediately after they are added, significantly reducing mutation rate during the replication process. This proofreading activity is shared by the DNA pole delta also. Thus, both of the DNA pole epsilon and delta ensure the minimum error or minimum rate of error during the DNA uh, replication process or throughout the DNA replication process. Now, the distinctive feature which separate the DNA pole epsilon from other primary DNA poles, that is pole alpha and pole delta, is well the distinctive feature which separates or which sets pole epsilon apart from pole delta and alpha is its high processivity activity and the P domain, which is this portion, which helps tightly to the DNA and thus increasing its processivity. DNA pole epsilon has an higher processivity even without the pCNA, but when it associates itself with pCNA, the uh, processivity increases several folds drastically. Now, the regulation mechanism is very similar to the other DNA poles, that is delta and epsilon, but the additionality is that cyclin-dependent kinases are also implicated in controlling DNA damage response pathways that intersect with the cell cycle checkpoints and thus indirectly regulate pole epsilon activity. Now, as I mentioned that the DNA pole epsilon primarily interacts with PCNA, where PCNA is the central cofactor for pole epsilon function during replication. It forms a sliding clamp around the DNA, allowing continuous synthesis by providing processivity to the polymerase complex. But PCNA itself can be modified post-translationally, that is, by or after the translation of the gene coding for uh, or mRNA which codes for the pCNA through processes like ubiquitination or phosphorylation, which affect its interaction with various proteins, including pole epsilon, and regulate replication dynamics. Now, the implications are one of the most important implication is genomic stability. Due to the proof-reading capability of pole epsilon, it plays a critical role in maintaining genomic stability by preventing mutation during DNA synthesis. Mutations or malfunctions within this polymerase can lead to replication stress and contribute to genome instability, which is associated with various diseases, including cancer. And every DNA pole image which I have used in this presentation is an real 3D model or PDB form or PDB model of the DNA polymerases of yeast cell, which I have downloaded from the official PDB site. Now let's talk about the processivity factors which play a role in the process of eukaryotic DNA replication. These are several proteins which play a crucial role in this mechanism by increasing the efficiency and fidelity of DNA synthesis process, meaning that it helps the DNA polymerase enzymes to synthesize continuous strands of polynucleotide chain without falling off the DNA strand. Here are some types of processivity factors, where some of them I have discussed in our previous slide. The first one is ORC complex. It acts uh, as an anchor site where other proteins involved in initiation and elongation can assemble. That is, when the initiation process takes place, the proteins like DNA pole epsilon, tCNA, clamp loader, and that is RFC anchors or binds itself to the or on the ORC complex. Now, the second one is CDC6, that is cell division cycle protein 6. It plays an essential role in controlling cell cycle progression through its interaction with cyclin-dependent kinase, as the process of eukaryotic DNA replication is tightly regulated with cell cycle. Thus, it also plays, or we can say that it also acts as an processivity factor. Then comes TDT1, which stands for cell division cycle, TF3C subunit, where our TF is transcription factor and this 3 indicates the type of RNA polymerase in which this factor acts on, that is RNA polymerase 3, which is responsible for the synthesis of tRNA. What it does is it helps in loading of other proteins involved in elongation phase like pCNA onto the uh, template strand photosynthesis by polymerase. That is, the CDT1 protein loads the pCNA onto the DNA strand or parent strand from which the DNA or newly synthesized DNA will form. And the next one is CDC7 and DBF4 protein. The complex formed between CDC7, a protein kinase that phosphorylates MCM proteins to activate them. As I mentioned previously, that there are several proteins which phosphorylate MCM complex in order to activate them, and CDC7 is one among them. And its cofactor is DBF4, which is essential in the initiation of DNA replication by promoting assembly of pre-replicative complex on origins, where DBF4 is essential for another kinase known as DDK kinase, which stands for DBF4 dependent kinase, which also phosphorylates some subunits of the pre-replicative complex in order for their assembly. The next processivity factor is MCM protein, which stands for mini chromosome maintenance helicase. As I mentioned, that it is an hexameric green protein. It plays a crucial role in unwinding the DNA so that the DNA poles can act on single strands and create a new double-stranded DNA in semi-conservative way. That is, using a parent strand synthesizes its complementary template strand. Thus, if the activity of helicase gets regulated, either down-regulation or up-regulation, then the entire process of DNA replication would be regulated. That is, the rate will change according to the regulation of MCM protein. The next one is pCNA, that is beta clamp and its different clamp loaders. And some of them are RFC1, DNA2 protein kinase, CSBF, BPC, PNKP, etc. Where PCNA is a sliding clamp that encircles the template strand and acts as an anchor for polymerase during elongation. Where, as you can see in this image, that this is the PCNA and this is the polymerase enzyme. Here, the polymerase epsilon. This is RFC, which stands for replication factor C. It loads the pCNA onto the double-stranded DNA, and the DNA polymerase gets anchored to the pCNA, which increases the processivity and fidelity of the DNA polymerases several fold by allowing the DNA polymerases to synthesize DNA continuously without getting dissociated or falling off from the DNA strand. How clamp loaders like RFC work is they uh, they help open up the PCNA, where PCNA in its initial unbound form is a closed ring. Thus, it cannot bind to the double-stranded DNA without an helping factor or any helper protein. And RFC acts as an helper protein, where RFC opens up the PCNA ring and loads onto the double-stranded DNA and then closes it. Thus, the PCNA clings to the double-stranded DNA. Now, the next possibility factor is topoisomerase. That is DNA. Some examples of topoisomerase are DNA2 endonuclease, topo2 alpha, chromatin remoter complex. What they does is they release the tension caused by the positive supercoiling due to the elongation phase or during the elongation phase of the DNA replication process by creating a single-stranded or double-stranded break, which creates a negative supercoiling, thus releasing the tension caused by the positive supercoiling. If they don't function properly, then the DNA replication process will get halted during the elongation process. Thus, the DNA replication process would never end or get stuck on a specific phase or a specific position. The next one is telomerase. Telomerase and a subunit of telomerase is TERT, which stands for telomerase reverse transcriptase. With HRP1 protein acts as a processivity factor. In eukaryotic cells that undergo multiple rounds of replication, telomeres shorten due to incomplete end replication by DNA polymerase enzymes. Well, we will talk about this particular point in detail in this upcoming slide in the topic of end replication problem. So, for now, just remember this point. Telomerase is an enzyme complex composed mainly of TERT and HRP1 protein as a processivity factor for elongation activity at chromosome end, thus maintaining telomere length in certain cell types like stem cells or cancerous cells. We'll discuss about this point in detail. Now let's talk about the coordination and regulation of lagging and synthesis, where the lagging strands is synthesized discontinuously as a series of short fragments known as Okazaki fragments. The entire process of Okazaki fragment and Okazaki fragment maturation can be described or divided into three portions. First one is primer synthesis, where DNA pol alpha primase synthesizes short RNA primers periodically on the lagging strand template to initiate Okazaki fragment. Then comes the fragment extension, where pole delta extends the primer efficiently aided by pCNA, form a discontinuous linear segment. And then comes fragment processing, where RNA primers are removed and DNA ligase seals the nick, and in place of RNA primers, DNA, or should we say that DNTPs are added, and then the nicks are filled by DNA ligase to produce a continuous strand. We'll discuss about each topic in detail in our upcoming slide. So let's talk about primer synthesis. We have discussed this uh, topic in far more detail in the topic of DNA polymerase alpha. But let me give you a brief overview for now. So, primers are synthesized by DNA pol alpha, primase subunit, especially by primase subunit, which synthesizes an RNA primer that binds to a single-stranded DNA. The purpose of this primer is to provide a free 3' hydroxyl group to the DNA polymerase delta or epsilon. As DNA pol delta and epsilon cannot synthesize DNA from scratch, they need a 3' hydroxyl group in order to extend the DNA strand. And this process is tightly regulated by protein interactions like pCNA and etc. The coordination of these processing steps ensure seamless integration of Okazaki fragments into growing lagging strands. This process also involves other factors such as RPA, which is a replication protein A, This is nothing but a single-stranded protein in eukaryotes, SSBP, uh, which prevents the re-annealing of denatured DNA and the clamp protocomplexes that help stabilize single-stranded DNA during synthesis. These fragment extension is basically the elongation phase of the DNA replication process, and we have discussed this also in our previous slide. So I'll also give an overview of this slide. That the leading strand is primarily synthesized by DNA pole epsilon, but sometimes DNA pole delta also synthesizes leading strand, and vice versa is true for lagging strand. This process occurs uh, repeatedly along the lagging strand as new Okazaki fragments are initiated. Processivity factor. The lagging strand is synthesized discontinuously because of its antiparallel orientation relative to replication force, that is the directionality of 5' to 3' and 3' to 5'. This necessitates repeated priming and extension cycles which are coordinated with leading strand elongation by cell cycle regulators such as cyclin-dependent kinases and CDK. Now let's discuss about the actual process of or mechanism behind the maturation of Okazaki fragment, which is also known as fragment process. Once the fragments are extended by at least one nucleotide past their primers, which have been removed and replaced with deoxyribonucleotides or DNTP, they must be joined together to form a continuous strand. This involves several steps, which are as follows. First step is the removal of RNA primer. There are various mechanisms or various enzymes involved in this process of removal of RNA primer. So first one is the DNA pole delta, which uses its 3' to 5' exonuclease activity to remove the RNA primers from the Okazaki fragment. But there are also specified enzymes for RNA primer removal, which gets executed by pCNA on pCNA itself. They are PEN1 and RNase H, as mentioned here. PEN1 stands for flap endonuclease 1, which removes RNA primer and any short overhanging flaps that may have formed during synthesis to ensure clean junction for ligation. Whereas RNase H is a special type of RNase enzyme which has high affinity towards a DNA-RNA hybrid, just like the DNA pole alpha, which also possesses a high affinity towards DNA-RNA hybrid. Thus, it is perfectly suited for the removal of RNA primer, as it only binds to the DNA-RNA hybrid formed by the primers and performs this exonuclease activity to cut down the primer. Here I must say that the RNase H uses its exonuclease activity to remove the RNA primers from the RNA DNA hybrid. So, and question arises that then why RNase H and PEN1 only degrade the RNA DNA hybrid of the Okazaki fragment? Why they don't degrade the RNA DNA hybrid which is formed during the transcription process? That is when a gene is transcribed by the RNA pole. There is a DNA RNA hybrid is formed even though for a transient time, but it is formed. Then why these RNAs H and FN1 enzymes don't degrade them, but only degrade the primers present in the Okazaki fragment during the DNA replication process? I would encourage you to pause this video and think about it for some time. So the answer is that the FEN1 recognizes the breaks between the Okazaki fragments in order to attach itself to the DNA-RNA hybrid. But whereas during the transcription process, there is no nicks or breaks in the strand. Thus, when one can't recognize the DNA-RNA hybrid present in the transcription or which is formed during the transcription process, whereas RNase-H cannot recognize and bind to the transcripted RNA due to the presence of 5' cap, as RNase-H is an exonuclease which removes nucleotides from 5' to 3' direction in order to cleave RNA from the DNA-RNA hybrid. So this is the mechanism by which flap endonuclease and RNase-H perform their activity. So when we see the activities of RNase-H, it firstly binds to the single-stranded overhangs or the RNA primers, then progressively degrades from 5' to 3', uh, uh, thus releasing free ribonucleotides, uh, which are which constituted the RNA primer. And after degrading the RNA primer and reaches the DNTP, it gets dissociated. And the activity of flap endonuclease is very is different than the RNA set. What it does is it recognizes either a flap or and single-stranded break. Then uh, cleaves cleaves the DNA and forms an flap-like structure like these. Then cuts the flap itself after some time. This is the activity of flap endonuclease enzyme. And as eukaryotic DNA pools like pole delta can degrade RNA primer and synthesize DNA simultaneously due to the presence of the exonuclease activity domain. Also, eukaryotic cells also possess pre-nuclear F1 and RNase H enzyme to some extent, which independently degrades the primers of the Okazaki fragment. As mentioned that the pCNA recruits the specialized enzymes like FEN1 and RNase H, but there are also some enzymes which are present inside the nucleus itself. Thus, they also uh, work independently to degrade the RNA primer. And after the removal and synthesis of RNA primers, the DNA ligase 1, 10, or 3 catalyzes the formation of phosphodiester bond between the adjacent Okazaki fragments, where there are multiple DNA polymerases are involved in the synthesis of gaps generated by the RNA polymerase, RNA primer, removal of RNA primer, but the most prominent one is DNA pol delta. Now let's understand the termination phase of the DNA replication process. The termination can be uh, divided into three major chunks, which are fork conversion and replication problem and telomerase activity, which we will discuss in detail in this upcoming slide, wherein fork conversion termination occurs when replication forks from adjacent origins meet and merge, completing DNA synthesis. Whereas in DNA replication and replication problem, chromosome ends cannot be fully replicated by conventional polymerases, risking progressive shortening. And the solution for the end replication problem is the telomerase activity. Telomerase is a specialized reverse transcriptase which extends the telomeric sequences, maintaining chromosome integrity over generation. Now we'll discuss each major subpoints or portions in detail. So the first one is force conversion. So to better visualize the mechanism, we'll take this image as a reference. So if we see that this is a replication bubble, this is an repliosome, this is an separate repliosome, and there are two replication forks in this replication bubble, and this is a separate replication fork. So what happens during the replication processes? One replication fork moves in this direction, and the other replication fork moves in this direction. These two are adjacent replication forks, which are continuously moving towards each other. So this repliosome is located on the top strand or bound to the top DNA strand, and this repliosome is bound to the bottom DNA strand. So what happens is when these two replication forks overlap each other due to a molecular cascade, the CMG helicase gets ubiquitinated, where the ubiquitination meaning a small peptide, namely ubiquitin, gets attached to the CMG complex. So after the ubiquitination, the entire repliosome will get dissociated from the DNA strand. Then the repliosome will disintegrate into its component parts, that is MCM27 helicase, genes, CDC5, CDC45, etc. So the key regulatory proteins are CDC45 and CDT41, which I mentioned in our previous slide. So these proteins ensure a controlled environment wherein DNA synthesis can be completed without interruption or error propagation at the convergence point. So now let's talk about fourth convergence checkpoint. So eukaryotic cells have built-in checkpoint mechanisms that monitor replication force progression and integrity, halting cell cycle progression if issues are detected during this critical phase of DNA synthesis termination. This ensures the fidelity of genome duplication before a cell enters meiosis or mitosis. So what happens is eukaryotic cells have a built-in checkpoint mechanism which continuously monitors the replication force progression and integrity. If any issues or mutations are there in the termination process, then the entire process will get altered until the problem is solved. During the phase of the DNA synthesis, this ensures the fidelity in genome duplication before the mitosis or meiosis. As we know that the DNA replication process takes place in the S phase. Now, the resolution after conversion. Once code convert and replication is complete, disassembly mechanism are activated, as I have mentioned uh, in or as I have discussed in this using this image. Now, the implications for genomic stability are post convergence is essential to prevent genomic instability as it ensures complete and accurate duplication before cell division occurs. Errors in this process can lead to mutations or chromosomal aberrations that contribute to diseases such as cancer or or any other genetic disease. Now let's move forward to replication problem, which is a fundamental issue that arises during the eukaryotic cell division due to the inherent limitations of DNA polymerase. So what happens is if we suppose this is the end of an chromosome. So what happens is during the synthesis of or during the process of DNA replication, this entire DNA strand will get converted into a single stranded DNA, then the DNA synthesis takes place. So this is an leading strand and this is a lagging strand. So the DNA pool alpha creates an RNA primer on the end of the uh, Okazaki fragment or lagging strand, which is in 5' direction. So after the maturation of the Okazaki fragment, as you can see that after the maturation of Okazaki fragment, we don't have any free 3' hydroxyl group for the DNA polymerases to perform their polymerization activity. So what happens is when this when the same DNA gets replicated again, the strand becomes shorter, shorter than the original one. Like, so what happens is when the Okazaki fragment is present, this is where this is suppose the end of the chromosome. DNA pole alpha comes in and creates an RNA primer at the lagging strand of the Okazaki at the lagging strand. So after the maturation of the Okazaki fragment, what happens is an void will be generated like this, or a gap will be generated like this at the end of the chromosome. And at the end, we have an 5' end, which is a phosphate group end. But an DNA polymerase an needs a free hydroxyl group in order to perform its polymerization activity. Thus, this gap remains in the chromosome even after the termination or completion of the DNA replication process. Thus, what happens is after some subsequent DNA replication of this specific strand or this specific strand, the DNA will or the chromosome will start to get shortened and continues to get shorter after each subsequent replication or cell division. So, in order to tackle this problem, our cells have developed an sophisticated enzyme which is known as telomerase, and we'll discuss about that in the next slide. Before getting to know about the telomerase enzyme, let's discuss some consequences of the end replication problem if it remained uh, unsolved. So first one is genomic instability. That is, as cell divides, critical genomic regions at the end become progressively shorter. This can lead to loss of essential genes or regulatory elements if not properly managed by the cellular mechanism like telomerase activity and alternative lengthening of the telomerase. So what happens is if the telomerase gets shortened subsequently during the cell's subsequent division, the newer cells will lack some essential genes which were present in the original parent cell due to the shortening of the chromosome or due to the end replication problem. So this results in the cell senescence, implications for cancer and impact on genetic diversity. Well, cell senescence happens due to the telomere shortening, which is associated with aging in eukaryotic organisms. And in modern days, there are many pharmaceutical companies are there which are developing anti-aging drugs targeting the telomere shortening problem. They are trying to, what you can say is, they're trying to uh, stop the shortening of telomeres in somatic cells like skin cells. Now, the implication of cancer. Some cancer cells bypass the normal replication termination and produces a large amount of telomerase enzyme, which continuously prevents the shortening of telomere. Thus, the cancer cells theoretically live forever. That's why we are still using one of the oldest human cell line, which is known as HeLa cell lines, which are cancerous cells derived from a human, namely Henrietta Lacks cervical cancer. And now the impact on genetic diversity is the end replication program can also influence genetic diversity within populations by affecting the survival of cells with shorter telomeres. That is, if a cell possesses shorter telomeres, then its future generation will undergo selective pressure due to their lack of essential genes, which we have discussed in the point of genomic instability. Now, the consequence of uh, telomere or sheltering complex that also plays role in the prevention of end replication problems. First one is the end protection. The T loop and the sheltering block ATM-ATR detection of chromosome end as DNA break. The length regulation, that is high TRF1, TRF2 protein occupancy on long telomere inhibits telomerase as telomere shortens. Reduced TRF1, TRF2 allows telomerase action. This process or this mechanism regulates the length of DNA so that the telomere doesn't lengthen too much or shorten too much. And there is the fusion prevention. RAP1 via TRF2 and FOT1 CPP1 suppress non-homologous anti-rejoining and homologous recombination at telomere so that uh, to prevent unnecessary DNA mutation which can cause uh, conditions like cancer. Now let's move on to the next slide. Now at molecular level, this is how a telomere would look like. This is the telomere in the chromosome, and this is its molecular zoomed print or zoomed snapshot, you can say. So this is the 5 prime region which I have mentioned during the explanation of the end replication problem, as this 5 prime end cannot be used by the DNA polymerase for the synthesis of DNA. So what happens is an enzyme only telomerase comes in and synthesizes the DNA, which we will discuss in detail. So for now, let's see the structure and composition of the telomere itself. So this is what we refer to as 3 prime overhang. So what happens is this 3 prime overhang invades an double stranded DNA upstream the telomere, thus creating a D loop which looks like this. This is what we refer to as D loop. And this overall loop which is formed in order to form the D loop is we refer to as T loop. This bigger loop is what we refer to as T loop, and the smaller loop is what we refer to as D loop. And these are the proteins which we altogether refer to as sheltering complex. And these TRF of proteins are what regulate the length of the telomere, as if the concentration of TRF protein increases, the telomere, the activity of telomerase gets reduced, and if the number of TRF gets reduced, the activity of telomerase gets increased. And the RAP1 proteins play a role in inhibiting the unwanted DNA recombination processes like non-homologous end joining or homologous DNA repair mechanism. Whereas these proteins, TIN, TPP, and FOT1 These proteins help in stabilizing the overall structure. Chain 1 binds to the double bound, so double-stranded DNA. The single strand of a double-stranded DNA. The TPP acts as a connecting link between pot and tin, whereas the pot binds with the single-stranded DNA of the D loop and stabilizes it so that it doesn't get re-annealed with the double-stranded DNA. Now let's talk about the telomerase enzyme in detail. So telomerase is the solution and a critical mechanism that addresses or solves the end replication problem, ensuring the chromosome stability and genomic integrity during eukaryotic DNA replication. This enzyme has two main components. First one is RNA component known as TERC, which stands for telomerase RNA component, which serves as a template, and the second one is protein component, which is known as TERT or HTERT, which stands for telomerase reverse transcriptase and human telomerase reverse transcriptase respectively. So the RNA component of the telomerase enzyme, that is TERT, acts as a template strand which is complementary to the telomeric sequence or repeat, that TTA triple G in humans, serving as a primer. So the third component acts as an you can say template for the synthesis of DNA by the TERT protein, that is human uh, or telomerase reverse transcriptase. What TERT does is it uses the RNA template, that is TERT, and synthesizes a complementary sequence of DNA, thus creating a unique or specific sequence at chromosome ends known as telomeres. And these telomeres are then decorated by various enzymes as I have mentioned earlier. Now the telomerase activity is telomerase selectively adds telomeric repeats onto the 3' end of the linear chromosome, compensating for any loss during replication and preventing progressive shortening with each cell division. This activity is tightly regulated in somatic cells to maintain genomic stability while allowing unlimited proliferation potential in stem cells and germline cells, where telomerase expression persists throughout the organism's life cycle or across generations respectively. So, expression of telomeres is especially high in uh, high rate proliferating cells like stem cells, germline cells, and in cancer cells. But in normal somatic cells, the telomerase, the gene which codes for the telomerase, is highly regulated. Thus, very low expression is there of telomerase enzyme. Now, the telomerase length regulation, which I have explained earlier, uh, is carried out by TRF protein. Now, the telomerase regulation is tightly regulated in somatic cells, as I said, through various mechanisms involving transcriptional and post-translational processes, including the binding of specific proteins, that is sheltering complex, to protect telomere from unscheduled DNA repair processes that could otherwise recognize the chromosome ends as sites for double-strand breaks or other form of genomic instability. And the protein which is responsible for that is RAP1. Now let's visualize the activity of telomerase. So this is the pictorial or graphical illustration of the entire process. This is the 3' overhang and 5' end of the telomere. This is the telomere enzyme. So what happens is telomere contains, this is the RNA component of the telomere. It contains the complementary sequence which can bind to the 3' overhang of the telomere. So as it binds, the telomere will synthesize an uh, DNA sequence which is complementary to this RNA template, thus extending the overall length of the 3' overhang. And after the extension, DNA RNA primer, or you can say the gap which is left behind by the telomere are filled by the DNA pole delta and ligase and DNA pole alpha, where DNA pole alpha will create primers and DNA pole delta will synthesize the DNA and remove primers, and DNA ligase will comes in and ligate any single strand breaks present in the telomere. This is this is also the same mechanism, but just in different uh, reference, if you can say that. And these are the references which I have used to create this presentation and video. And thank you for watching.